Hello. My name is Sam, and I hope to get feedback from you on how to improve this talk. Right. So, my title is DNA is all you need. It's very surprising how, from a single genome, you could get this transcript or RNA and produce these proteins that really plays a role, maybe like in movement, nutrition and metabolism, and also reproduction. So there's a lot of information you could get from the genome, right? But the genome is also highly regulated, and one of the mechanisms is called epigenetic regulation of epigenetic regulation of DNA. And what I'm really focusing on is methylation. So it's basically tags on the DNA where when they are methylated DNA, it becomes really difficult for transcripts to be formed. And in regions of the gene where there is less methylation, transcription goes on, and then you could produce the proteins that could generate activities of life. There are other factors that really regulate gene expression, like transcription factor binding, and also the 3D and 4D genome organization. So the goal of this project is to develop a model that would predict gene expression from methylation data. And the experiment is set up in this way. So we have this machine called the Oxford Nanopore, which basically gives you a readout of the DNA. But in addition to that, you could get methylation information as well. And what we did was to take some mice cells and basically sequence them using this machine, and then do something called the RNA sequencing, which is basically measuring the amount of transcripts. But then the goal is to generate sort of like a computational model that would skip the RNA sequencing part. This is how the data is organized on the far right, and this is how the data is when I put it in the computational model. So I created generative model. Basically, it's a model. Don't worry, I'm also panicking by the mathematics. But basically, it's a function, right? So a function is a machine where you put in an input and then you get an output, okay? And then in this situation, the input is the methylation information, but then we sort of like do some summary statistics of this value, and the kind of summary that we did was to calculate something called entropy, which shows how disorderly methylation is at that particular gene. And then we put it into this machine, which is a function, and the function definition is this. But just think of this as this. And then what we get later on is the RNA values. So this is also how to represent this information in sort of like a box, plates in a diagram way. So how well did we perform? Right? So you have this computational, model or machine, where you put in inputs and get some outputs. So it means you're not limited, by data, right? So you could use this model to generate sort of like synthetic data sets and see how best you are performing compared to the real results. So this is called the prior predictive, samples, and then the blue dot. And then the blue dot is the actual, true values. So the DNA entropy and then the corresponding RNA value. And then the blue dots are actual values. But then the purple values are synthetic data sets we generated from this model. So this is one, the model hasn't seen like the real data yet. But then we could see that the synthetic data sets, it's on the right, sort of like centered around, the true data set is centered around the synthetic data sets. And this is when you run the model and then you condition it on actual data set. What I can see is that our model sort of like underestimates the true RNA expression. So there's room for improvement, but then this is sort of like the framework of how we are doing things. So I think one of the things I'm trying to do in the future is to sort of like, instead of predicting for one particular gene, is to pull all the genes and all the samples together and then do something like a pooled analysis and use another sort of like equations to be able to get the results that we're looking at for. Another thing I'm also trying to do is to use other summary statistics. So what I showed you was using the entropy, or we could use other things as well, like the geometric mean, the mean of DNA methylation, and also the median of DNA methylation. With that, I say thank you.
Thank you. Questions?
So with this model,
Here we go.
People online.
This is kind of a specific question, but with this model, what is the benefit of doing this? Or like, do you think that this would be combined with doing something like Hi-seq or ATAC-seq or ChIP-seq in order to look at the epigenetic profiles? Like, what makes this unique? Is this like for high throughput for like patient samples? Or why wouldn't we just go for the methods that currently exist?
Well, I mean, so basically this, if this is successful, this will save us a lot of money because you need special instrumentation to do RNA-seq. But if you have sort of like a complementary sort of like computational model to predict RNA just from DNA, I think that would be huge cost saving. And also to predict other things like transposable, to study transposable elements very well. So I think it will be complementary if it's successful. That's my opinion. And then the limitations of this is that it doesn't sort of like characterize sort of like other modalities of gene regulation, like things that happen at the enhanced regions. Yeah.
Thank you, Sam, for this talk. And I think my question kind of plays on what she asked and to your answer, that it would save costs if your model is successful. And I think over time, like during my work and my study in other African countries in their computational and bioinformatics department, that's one thing that's a challenge, like finding ways to save costs, to be able to look at these samples, to be able to build these models, because frankly, they do not have the manpower and also like just the funding that we have here. So I think it's very amazing that you're doing this work and it can be actualized, not just here within the Tri-I, but outside of our institution and America as a whole. So thank you. My name is Saramdo Tete. I am at MSK in the Solid Lab, but I will be starting a PhD in bioinformatics at NYU in the fall.
Thank you.
Thank you. And as one who's in global health, I think the issue of money between the global north and the global south, who sets the agendas, who decides what's important, gets back to this, because if you have all of that infrastructure, then you may need other ways to be able to do it, and you're more alert to that, as well as some of the movement they have now to try to make sure that there is more of a transfer of that infrastructure. And I don't know how many of you know, but one of the issues in the fields are that we have people who train very highly here, and then their skills are needed back home, if you will. But when they get there, they don't have all of the equipment, they don't have all of the infrastructure, and so in many ways that investment, that seed, cannot proliferate and grow. And so one of the things that we do, again, with the Society, the American Society of Child and Hygiene, is we have many members that are from LMICs, or lower middle income countries, is to actually have to talk about how can you sustain your career when you return home, or even if you want to stay here and you want to continue to work. And as COVID-19 showed, we really need to be able to have better work across these borders to get the sort of interdisciplinary teams and understand what's going on, and to have the sorts of analyses. So kudos to you for what you're doing, Sam, and good luck to you at NYU. Remember, you already got home up here, so. Oh, okay, she'll still be here, so we're going to have you come and speak next time. Okay, thank you so much, Sam. Thank you. Can we give a quick cheers? And Nicholas is next. Nick.